Hi everyone, my name is Alana Kalehua and I hope you're having a good day. We're going to learn about some studies done by Thomas Jessel and his colleagues showing that ectodermal BMP induces dorsal fate. We're going to be learning about some experiments done in a paper entitled Dorsal Differentiation of Neural Plate Cells Induced by BMP-Mediated Signals from Epidermal Ectoderm. For some quick background, there were a few things already known about cell differentiation during development before this was done. Thomas Jessel and his colleagues were the ones who discovered that sonic hedgehog was the secreted morphogen that provides a ventralizing signal. And it has also been learned that while the notochord is not required for dorsal differentiation, if the notochord isn't there, genes that are typically restricted to the dorsal regions will expand to all dorsoventral levels. So this raises the question of, are dorsal fates the default fates, or is there a dorsalizing signal just as there is a ventralizing signal, which is sonic hedgehog? And a couple past studies on neural crest cells have supported the evidence of dorsalizing signals, and those authors suggested looking into epidermal, ectoderm, and mesodermal cells as possible contenders, which these authors do too. We'll come back to that in a moment. And so these authors begin tackling the question of how does dorsal differentiation occur by some in vitro assays with neural plate explants. So the first thing these authors did is they looked at 4 genes that are typically expressed in the dorsal part of the neural tube, and they're asking, is dorsal fate the default thing, or other inductive signals also required? So they looked into 4 genes, MSX1, PAX3, DSL1, and SLUG. And what they found was that MSX and PAX3 were restricted to the dorsal regions of the closed tube in expression. And some previous studies have also shown that the notochord can repress their expression. So these authors concluded that PAX3 and MSX are initially expressed uniformly along the neural plate and then are subsequently repressed to just the dorsal regions by the ventral notochord signals. So they are not sufficient enough to dorsalize tissue all on their own. Next, these genes were continued on to the next stage of the experiment, where they were used as markers in explants taken from prospective dorsal, intermediate, and ventral regions of the neural plate isolated from chick embryos at 10 days. And so they were isolated and grown without notochord signals. And the question that these authors were wondering is, what's going to happen in each of these regions? So the prospective dorsal tissue differentiated into dorsal tissue, the ventral tissue differentiated into ventral cell types, and the intermediate tissue didn't really do either. So this shows that while the notochord isn't going to prevent dorsal differentiation, it also isn't going to be enough to just have a missing notochord. There also are other signals required for dorsal differentiation. And now we return to the suggestions of the previous authors in earlier studies, looking at where these dorsalizing signals could be coming from. These authors looked into working with epidermal ectoderm and periaxial mesoderm, and they took ventral neural plate explants and grew them in contact with these 2 substances. The explant that was grown with the mesoderm had no dorsal induction. However, the neural plate explant grown next to the epidermal ectoderm did have neural crest cells migrating from it, which shows that epidermal ectoderm is the source of the contact-dependent dorsalizing signal that these authors were looking for. Now the next question is, what exact molecule is coming out of the epidermal ectoderm? And here's where we bring BMPs into play. So previous studies had shown that DSL was in the family around dorsalization, and so maybe some of its other family members could also be in play here. And so a PCR analysis was run to isolate structurally related genes that work similarly to DSL that could be possible contenders here. And the PCR suggested they look into BMP4, BMP5, and BMP7. And the authors found that BMP4 and BMP7 were expressed in the right area of the neural plate, so they continued working with those. But the question is, are these BMPs actually causing the dorsal differentiation? So in order to determine this, BMP4 and BMP7 cDNA expression vectors were transfected into COS cells, and they induced neural crest cells, showing that BMP4 and BMP7 can induce the dorsalizing factors that they were looking for. To wrap this all up, these authors were initially looking for how dorsal differentiation occurs and what affects it, and they were able to find that there are some genes that are involved in dorsal differentiation. However, they are not enough. There are still some localized inductive signals that are required. And there are three main conclusions that they derived. The first is that the epidermal ectoderm that flanks the lateral border of the neural plate represents the source of dorsalizing neural plate cells. And second, the molecules BMP4 and BMP7 are expressed in the epidermal ectoderm, and they mimic the dorsalizing activity of just epidermal ectoderm. And to wrap these findings up into the broader picture of things, Thomas Dressel and his colleagues had previously shown that sonic hedgehog was a ventralizing factor from the notochord, and here they show that BMPs are dorsalizing factors coming from the epidermal ectoderm. So the differentiation along the dorsal ventral pathway is likely mediated by opponent activities of BMPs and sonic hedgehog. Thank you so much. Have a great day.